Okay, so for the next experiment, I'm going to talk about how you can actually measure the rate of photosynthesis. All right, so you should remember that photosynthesis uses light energy and oxygen and water to produce carbon dioxide and glucose. All right, so I'm going to just put the equation down for that. Okay, through the magic of pausing video. So this is your balanced chemical equation for photosynthesis, right? So this is your CO2, this is your water, this is your glucose, okay, and this is the oxygen that's produced. And so if you want to directly measure how much photosynthesis is happening, the rate at which it's happening, how quickly it's happening, or how much it's happening, it can be a little difficult to do with land plants because the things that are being produced are glucose, which is hard to see, as well as oxygen, which is a gas. So in land plants, that's diffusing out of the stomata. But we can observe it using a underwater plant called pondweed. Okay, and so the idea for this is that if you have a beaker, you guys know my awesome drawings, but there's your beaker, okay, and it's filled with water, and then the pondweed sits in the water. This is exactly what it looks like. Truly authentic drawing. Okay, so that's my pondweed, okay, and it's under the water, and then you shine a light on it. Right? That's my light source. Okay, and so the light will allow the pondweed to do photosynthesis. And so you can measure how quickly that's happening by looking at the production of the oxygen gas, because the oxygen gas will be released from the leaves or the stem and as tiny little bubbles that will pass up and rise up. And so you can count, the idea is that if you count how many bubbles there are, then that tells you how quickly over a certain period of time, right? So you can count the number of bubbles in a minute or in 30 seconds or whatever. And so you count the number of bubbles, and that tells you how quickly photosynthesis is happening. So you could adjust the light, for example. You could put a thermometer in if you wanted and adjust the temperature of the water, see how that affects the rate of photosynthesis. You could provide carbon dioxide in here. You could put a chemical in that provides carbon dioxide, and you could mess around with the concentration of that. You could mess around with the temperature, the light intensity, all of these things that affect the rate of photosynthesis. All right. So counting the number of bubbles is the usual output for this, usually the dependent variable. But if you have a method of doing this, you can actually improve on, rather than counting the number of bubbles, you can actually capture the gas and measure the volume that is produced, okay? Because hopefully you can imagine that these bubbles may be different sizes, and so they could actually have different volumes. So counting the number of bubbles is perhaps more qualitative than quantitative. And so if you want to do it more quantitatively, you would capture the oxygen and measure the volume. And the way you would do that is, so for example, you could put a test tube or measuring cylinder over the plant, all right? And then if that has markings on it, you could, for measuring the volume, then you could actually, the idea is that the bubbles, let me get my color for the bubbles here, would actually be trapped up here. And so this would be your oxygen gas. And then you could literally see how much has been produced. And then if you time it, right, so if you take your volume and you divide it by the time that it's taken to produce that, then you get your rate of photosynthesis. Remember, okay, that when you're doing this, right, the rate of photosynthesis, so it's not just about how many bubbles are produced in total, it's about the number of bubbles or the volume divided by the time over which you are recording your data.